So, one, two, three, go. So, good morning, everyone. So, we are the group three, and we are here to discuss to you our assigned report, which is about the characterization of microbial communities under the course Microbial Ecology from lecture 7 to 9, which will be discussed to you by Baranda, Binan, Ludrieta, Sale, Surigao, and yours truly, Villegas. So, as a introduction, we all know that microbial characterization is very essential part, in studying microorganisms. So, and also, microbial characterization also plays a pivotal role in understanding the ecological system and interaction. And by characterization, through characterizing microorganisms, we could elucidate them, their unique traits or behaviors, or some of their physiological capabilities or metabolism that shape their interaction within the ecosystem. And also, this also give us insight, through their characteristics on how microbial diversity influences ecological balance, nutrient cycling, and environmental resilience. Next slide, please. Wait. So here are the content of our lectures. No, so I will be discussing first the culture-based methods, and the rest of the topics will be discussed by my fellow group mates. So next slide, please. As an introduction to my report, assigned report, um, we could say that there is a saying, or there's a count that says that the mass majority of microorganisms, so with a percentage around more than 99% of all species by most estimates, have never been grown in laboratory culture. So imagine around negative around less than 1% of the total population of microorganisms have never been cultured, or have been already cultured, and the 99% of it are in still in discovery pa. So we could say here that there is a huge gap pa in the knowledge about microorganisms in terms of their characteristics, and we could say that there is a lot to be discovered pa in the field of microbiology. And to date, around 17,000 pa lang of archaea and bacteria have already been formally described. So in a laboratory setting in this context. So however, by contrast, through the advent of technology, na natay mga cultural independent na mga methods, which um, mostly involves molecular technique, and the molecular diversity survey says that it indicate that if many millions or even trillions pa na estimate nila na around millions or trillions pa of species exist in nature that need na ano pa unidentified pa, or need to be cultured pa or formally described pa. So we could say here that the culture based method is has a limitation pa, kay knowing na ang percentage pa lang sa up to now is very low pa, which is around 1% pa lang ang na-discover, na grow sa laboratory culture. And but still the culture media isolation or culture media characterization is still or remains the only way we could fully characterize its properties and predict its impact on its environment. So wala pa ring, there is no substitute pa sa enculturing, um, in a culture-based method, uh, than the other way and then the other mga ways para ma-characterize ang microorganism. So and also there's a term here na candidatus. So taxonomically speaking, these are the microorganisms that cannot yet be cultured in the lab but are identified through molecular methods. So there are some na species of microorganism na ganito ang kanilang estado, which is they are fully characterized through molecular methods, but in a culture-based method, hindi sila pwedeng or hindi feasible ang pag-culture sa kanila given the fact uh, na may mga kailangan na mga environment na need to be achieved para mag-grow sila fully. Next slide please. So now I will discuss to you the culture-based methods. Next slide. So once we are once na gusto nato mag-characterize a microbial communities through culture dependent analysis, one of the first na mga methods that we will be going to employ is the enrichment culture. So sa enrichment culture, it consists of a medium and it is followed by a set of incubation conditions. So which this type of medium and incubation condition are established that are selective for the desired organism. So in other words, it this only provides an environment that promotes the growth of only the target organism, and it is also counter selective for undesired organisms. So it inhibits the growth of other undesired organism na mag tubo sa medium or sa enrichment culture. So, and one of the important consideration once we are trying to enrich a microbe in a culture media is we should be able to duplicate as closely as possible the resources, conditions of a particular ecological need. So there is a necessity na kailangan natin mamimic ang ang environment or masat ang environment sa enrichment culture sa kanilang natural habitat for us to offer the best chance of obtaining the organism of interest. So there are three kailangan na aspects na need to be na need to take consideration. First is the resources and nutrients. So dapat we should aware about the nutrients na need sa target natin na microorganisms that as nutrients provide the essential building blocks for microbial growth and energy. Next is the incubation time. So important, this is also one important aspect na i-consider natin, which proper incubation settings so such as yung kadugay or duration and stability are crucial for the consistent environment. So we are promoting here for a consistent environment that supports the organism's growth cycle. And next is the condition. So there are a lot of condition that needs to take consideration. So sa temperature, sa pH level, osmotic consideration, and also we need to determine kung whether it could be aerobic or anaerobic microorganisms and the like. Next slide, please. S- So, as I said a while ago, one of the key aspect for enrichment culture is the media. So, as an example here is about I'm set an example po, mag-set po ako example. So, if we try to isolate or ang subject interest nato is halophiles, mahalophiles na mga microorganism, ang media, one of the aspect na need i-consider that is ang media. So one of it is ang kanyang um example here is the broth, which is it intended for the extreme halophilic species of Halobacterium and Halococcus from foods. So here, as we can see here na notable ang sodium chloride in terms of the quantity of grams per liter because this type of microorganism require a high salt concentration of about 20 to 30% of sodium chloride. So in this type of media, we could have a what we call on assurance na once na mag-inoculate tayo ng ating target organism here, which is for example halophiles, we could have a assurance na obtain tayo nun na type of microorganism. Next slide please. Next slide. And also one temperature is also a consideration po for setting an enrichment media. So there are a lot of types of microorganisms that reside in an optimal conditions. So in the psychrophiles, around -10 to 4 degrees Celsius ang kanilang optimal temperature. Sa psychrotrophs such as the E. coli, 10 to 39 degrees Celsius, mesophiles, around 40 to 60 degrees Celsius, 8 thermophiles such as the Thermococcus scalar, around 60 to 88 degrees Celsius, and the hyperthermophiles around 90 degrees Celsius and above. So temperature is very crucial once we um, once we are try to manipulate or to create our enrichment media. Next slide, please. Next aspect na need to take consideration is the location. So successful enrichment requires an appropriate inoculum containing the organism of interest. So here in the table showing the some enrichment culture method for phototropic bacteria. So these are the bacteria na they are using light energy as their a light source as their energy. So um, if we want to enrich cyanobacteria, um, the they are typically found in pond, lake, water, sulfide-rich muds, and etc. And also sa thermophilic cyanobacteria, no, if we want to enrich that, or it is our subject of interest, they could find in hot spring microbial mat. And then sa mga anoxic na mga organisms such as the purple non-sulfur bacteria, heliobacteria, um, they could be found in the hypolimnetic lake water, pasteurized soil, microbial mats for thermophilic species. Next slide, please. Next slide po. And now, and continuation po for other bacteria such as if we want to ang mga target is non-phototropic bacteria, so they do not use energy or light as their energy source. So previous slide pa. Next, number 10. Yan. So if you want sa mga chemoorganotrophs na aerobes na mga microorganisms such as Mycobacterium, Nocordia, Azotobacter, Cytophaga, they could be found in soil, mud, lake sediments, decaying vegetation, and thermocline of certified lake. And gusto natin sa mga chemoorganotropes na anaerobic respiration and the utilized fermentation such as Pseudomonas, Bacillus, Desulfomonas, they could be found in soil, mud, sediment, sewage sludge, and many more. Next slide, please. And those sa mga bacteria na chemolithotropes, so meaning they use inorganic components as their energy source, they could be found in like sediments, thermocline, stratified lake, acid mine drainage sa mga aerobes such as nitrite oxidating bacteria, hydrogen bacteria, Theobacillus, acid, Theobacillus and many more. And sa mga anaerobes, sa mga microorganism na chemolithotropes such as Geobacter, Theobacillus denitrificans, Paracoccus denitrificans, methanogens, they could be found in groundwater, mud, lake sediments, soil, and many more. And to and therefore the making of enrichment cultures begins with collecting a sample from the appropriate habitat to serve as the inoculum. So very essential ang location na ating kukunin to obtain the subject of interest na microorganism. Next slide please. So another form of enrichment process is the Winogradsky column. Previous slide po. So Winograd column. So Winograd column is an artificial microbial ecosystem and a long-term source of various bacteria for enrichment culture. So Winograd column is prepared by first filling a glass cylinder about half full with organic rich, preferably sulfidic mud into which carbon substrate has been mixed. And typically, ang mud is supplemented with small amounts of calcium carbonate as a buffer and gypsum as a source of sulfate. And the top layer is covered with foil to prevent evaporation, and the container is placed near a window that receive a sunlight for a period of months. And later on, we could observe coloration sa different layers of the column. And here in sa figure, we could see here that um, every layer of the column portrays a different microbial environment. So at the topmost layer, we could observe here na there is a high concentration level of oxygen, and once na magbaba in depth is nag-decrease ang concentration sa oxygen, and which this part of the topmost layer is considered as the oxic. So meaning plenty ang presence of oxygen. And in contrast naman sa bottom layer, we could see here na ang H2S is high in concentration in the bottom, and then nagpa-decrease siya when gearing towards the middle part. And in the bottom part, um, it is a layer in which anoxic environment is observed, and in the middle part is suboxic. And so topmost part is this ang mga microorganism na present here are the algae and cyanobacteria, which they quickly develop in the upper portion of the water column, producing oxygen and help this zone of the column oxic. And while sa next part naman is as the level of oxygen concentration decrease, facultative anaerobes such as purple non-sulfur bacteria grow. And these bacteria can perform photosynthesis without producing oxygen. So that's why they are facultative anaerobe. So meaning they can switch to aerobe and anaerobe state. And then they can perform photosynthesis without producing oxygen by using only light and available organic compounds for energy. The next one is the sulfate reducing bacteria and oxidizing bacteria. So these are, because in the figure, ang layer is the patches of purple sulfur or green sulfur bacteria such as chromatium or chlorobium, and they use sulfide as an electron donor for photosynthesis in low light. And sa bottom part, which we could makikita natin dito ang mga strict anaerobes including fermentative bacteria which decompose organic matter producing byproducts like organic acid, hydrogen, which further fuel the sulfur cycle. Next slide, please. Next slide. Now once na naka-create na tayo ng enrichment media, hala, previous pa. Previous pa, please. Previous po. The Angel, previous po. Previous te, so wala pa na te. Previous. Yan. Wala. Okay na. Ano? Okay na. Okay, so once na naka-establish na tayo ng enrichment media, so the next step is the isolation method. So these are the classical procedure for isolating microbes. So it could be done through strict plate, agar dilution, and liquid dilution. So in strict plate, so it typically use a agar medium, and its principle is it aims to spread microbes on agar plate to spread or to separate individual colonies to attain a isolated colonies, and it's considered as a quick, easy, and method of choice. And if a well isolated colony is selected and restricted several successive times, a pure culture is usually obtained. Next is the agar dilution. So it is a mixed culture diluted in tube of molten agar medium, resulting in colonies embedded in the agar. No, as we can see in the picture na may mga isolated colonies embedded in the molten agar, and it is done by serial dilution, and the target microorganism here are the obligate anaerobe such as phototrophic sulfur bacteria and sulfate reducing bacteria. And next one is the liquid dilution rather. So serial dilution is um, conducted here of an inoculum in a liquid medium until the final tube in the series shows no growth. And besides being a method for obtaining pure culture, serial dilution under the liquid dilution are widely used to estimate viable cell numbers in the most probable MPN or MPN technique, which is ideally much more efficient sa if replicate ang type of pag every tube is replicated, so meaning it is more preferable na efficient ang result na ma-obtain. Next slide please. Previous slide. Te. Ala, te, sobraan na ka te. Previous pa. Okay. Now, so here are the criteria for culture purity. So from the enrichment media from the method of enrichment media to the isolation, now ang main goal naman is for obtaining a pure colony. So these are the criteria that needs to be considered needs to take into consideration to have no to purify or to have a pure culture. So first is microscopy. So through the methods of gram staining, we could examine cell morphology and uniformity of staining. Next is the observation. Through its morphological characteristic or colony characteristics, and which we could assess here the colony appearance on plates or in dilution tube for consistency. Next is it could subject the growth into a various media such as selective media, differential media, and it is important na para ma-further classify pa ang microbes na subject of interest natin or need natin i-characterize. So in the next slide please, is some of the examples na methods para ma-further classify pa ang ating subject of interest or ang ating microbes through various media. So first is yung blood plate agar or BPA. So it contains the sheep blood allowing growth for fastidious organisms and differentiate bacteria based on hemolytic activity. So it considered as blood plate agar is considered as a enrichment media and also a differential media. And some example of it is sa Salmonella species na strict sa blood plate agar, we could see na very grayish ang color niya and there is no hemolytic activity. In contrast sa Staphylococcus aureus, we could see here na may mga spread na around colony may mga dense na color which serve as the sign as na may hemolytic activity ang isa ka organism. Which sa E. coli, we could serve na parang same lang sila sa Salmonella, which once na inoculated sila sa blood plate agar, they don't have a hemolytic activity. Next slide, please. Next slide, please, is this slide is nutrient agar. So nutrient agar is considered as a general media. So it supports the growth of wide variety of non-fastidious organism without sensitivity or differential features. So we could observe here na parang same lang sila ng colony, ng color, which is parang opaque or um, transparent in color, and media or agar media is there's no differential method na or differential na mga characteristic na ma-observe, so that's why uniform lang sila. Next slide please. Next is the previous pa. Okay. Next slide. Next media is this XLD or Xylus lysine deoxycholate agar. So it is a selective media na only gram-negative bacteria can only grow, and it differentiate based on carbohydrate fermentation and hydrogen sulfide production. So this type of media uh, only is a it is a type of differential media which only select gram negative, and mga gram positive bacteria once mag-inoculate kayo dito na type of media, there you could observe no growth. And sa salmonella species, ang ma-observe natin is may mga black colonies siya, which indicating na H2S, nag-produce sila ng H2S. On the other hand, sa E. coli, there is no black centers ma-observe, so meaning there is no, wala sila produce ng H2S, but they ferment xylose, so resulting in yellow colonies. So this type of media also, aside from differential siya, aside from selective media siya, differential media din ang type niya. Next slide please. Next slide. Previous slide pa. Previous slide pa. Previous slide. Pati. Okay, ito na. Next is the EMB, agar, or eosin methylene blue, and it is also a selective media. Only gram-negative bacteria can grow, and it differentiates based on the lactose fermentation. So, in Salmonella, it is a non-lactose fermenter, so producing clear lang and light purple colonies. However, sa E. coli, it has a strong lactose fermentation leads to metallic sheen green on colonies. So we can observe here na it was also indicator din na once na ang inoculum mo is may E. coli, once na kung mag-strict ka sa EMB, may ma-observe ka na metallic sheen green, and that metallic sheen green indicator na may E. coli ka na strict. Next slide please. Then next, the hectowin enteric agar. Selective pa rin siya sa mga gram-negative bacteria, and gram-positive hindi makagrow. And it differentiate based on lactose fermentation and hydrogen sulfide production. So kung non-lactose fermenter, there is no um, color change, and green colonies with black centers production are observed in Salmonella. Pero kung kung ang bacteria is nag-ferment ng lactose, it producing yellow to salmon colored colonies, and mag-change din ang medium from blue to red. Next slide please. And now na once na na-stick natin ang mga isolated from differential media, no, na further classify pa natin siya, the final evidence for culture purity is, first is uniform cell morphology understanding, no, under once we observe in microscope, and also consistent colony characteristics across growth media, and the most important is the lack of contamination in different growth media tests, para pure lang ang, para ma-ensure natin na one type of species lang at a time ating ma-characterize every plate. And once na, if we want to try to verify pa ang culture purity, dito na magpapasok ang mga molecular techniques. And however, it said here na these methods complement only but do not replace the traditional observation of cultural characteristic and cell morphology of a microbe. So in the next slide, my fellow groupmates will discuss to you more on about the molecular methods or the non-culture based methods in characterizing microorganism. That's all. Thank you.
Hello, good morning everyone. Marinig ako. Okay, thank you. So, my name is Angel May Binyan, and I will be discussing the uh, characterization of microbial communities using biomarkers and cell states. Sa ba si Moises? Ay, sorry, galag akong laptop and hinay ang connection dere. So here are the objectives of my discussion. So I have five goals in this discussion. So first is to explore the role of biomarkers and cell stains in characterizing microbial communities. Second, to classify the different types of cell stains used in microbial characterization. Third, to examine the application of fluorescent dyes as biomarkers for microbial cells. Fourth, to assess cell viability in microbial communities using viability stains. And lastly, to evaluate the advantages and limitations of these biomarkers and cell stains in microbial ecology. Ay, kalags. So, why study microbial communities? So, na-discuss na ito ni Patrick a while ago, but allow me to repeat it. So, we have five importance or uh, for studying microbial communities. So, first is it helps us understand this ecosystem balance since we all know that microbes play a crucial role in mostly in every nutrient cycling, such as carbon cycle and uh, the ano ba to? The nitrification whatsoever, and then it helps in giving us insights in the health and disease. So studying these microbial communities can help us understand how microbial imbalances leads to diseases and how to manage or prevent them. For example, in the gut. So we all know that the gut hosts diverse microbial communities, and Sha- uh, this includes the good and bad bacteria. So changes or sudden changes rather in this microbial uh, community balance in the gut indicates that there is a digestive health problem or disease in the gut. Next is the environmental impact. So it helps us understand their contribution to environmental processes and how they could be used in bioremediation. And next is industrial application. So these microbial communities are widely used in industries such as agriculture, food production, and biotechnology. Lastly, is it helps us gain insights about the evolution of ano, genetic transfer resistant mechanisms and how organisms adapt to new challenges, which is critical for fields like medicine, agriculture, and ecology. So, why study biomarkers and cell stains? So, by using these tools, which is the microbial staining and biomarkers, we can identify the microbes involved in biological processes such as nutrient cycling, and this give this help us track their population changes and understand their impact on ecosystem and human health. So, what is biomarker? So, biomarker, short for biological marker, is an objective measure that captures what is happening in a cell or an organism at a given moment. So, in microbiology, biomarkers are specific biomolecules that uh, can be proteins, lipids, nucleic acid, genes, in even a whole microbial species that can reveal information about the uh, microbial community. So they are often used to quantify microbial populations in various environments. So these biomarkers can serve as early warning systems for health and organisms. Hala, wait lang, sobraan. So example scenario. So first of all, biomarker was first used in the field of medicine. So in early 1800s to mid 19s, approximately 100 years, exploratory surgery was utilized kasi uh, to ano to to diagnose certain diseases. So this approach allowed physicians to directly view and assess the condition of internal organs for centuries. It was one of the only ways to confirm diagnosis related to internal bleeding, tumors, and abnormalities, especially in abdominal cavity. So before biomarker was discovered, so classical diagnosis was the exploratory surgery. For an example scenario, here is a guy, a person, experiencing a severe stomach pain. So, hindi na niya kaya yung sakit na nararamdaman niya. Uh, this person, this guy, will visit hospital, of course, to see a doctor. So, the doctor will hear out these complaints of this man, and then from further discussions and further agreements will lead to exploratory surgery para makita mismo or ma-confirm if nasa abdominal cavity yung uh, deceased or ano ba, masakit lang yung tiyan niya. So, ganon. That's how um, it was diagnosed na may something wrong with the gut or the stomach in early 1800s to mid 19th. So no utilization of biomarkers. So so of course, this classical diagnosis or the exploratory surgery has disadvantages and advantages. So few advantages includes the direct visualization. So of course, this could identify conditions that could not be assessed externally kasi open surgery will be performed in the organ or body mismo ng patient. And then, next is the definitive diagnosis. So, for conditions with ambiguous symptoms, often provided a conclusive diagnosis, especially for detecting tumors, internal bleeding, or organ damage. Next is immediate intervention. So, in cases where a life-threatening condition was discovered during surgery, surgeons could address the issue immediately without needing a second procedure. Next and lastly is it is broadly applicable. So, it is available for a wide range of internal issues across different organs and body systems. Oh, okay. So here are its disadvantages. So first is its high risk kasi um we are all aware naman siguro na hindi pa high-tech yung mga gamit in early 1800s to 1900s. So there are classical aseptic techniques used which could lead to high risk of infection, blood loss, and complications from anesthesia. Next is invasiveness. So, since it is an open surgery, it was highly invasive and required a longer recovery period, which could increase the burden of the patients. Next is limited accessibility. So, this approach requires trained surgeons and fully equipped facilities, which were not always available during that times. Especially, it is costly to be a trained surgeon, and not all can uh, acquire this privilege of uh, being fully equipped facility in hospitals. Next is high cost. So it is often costly in terms of time, resources, post-operative care, which limits its feasibility as a routine diagnostic tool, particularly for patients without immediate health insurance or financial support. Um, Next and lastly is the lack of precision. So it allows direct viewing. However, it could not always reveal certain microscopic or molecular details about diseases such as infections or small diffuse cancer cells, which can be detected through advanced imaging and molecular testing, which was not invented during this era. So when these biomarkers and cell stains are discovered, which is during this modern times, so this exploratory surgery was being replaced by non-invasive technology that allows detailed analysis of the body's internal state without the need for open surgery. So, one of the most significant advancements in is the use of molecular markers and cell staining techniques. So, these techniques allow safer, faster, and more precise ways to diagnose diseases, which enables doctors to detect cellular and molecular changes at early stages and understand disease mechanism at much deeper level. So again, in the same scenario, a person having severe stomach pain goes to an hospital and then consults to a doctor. So the doctor will hear out the complaint and the doctor will get blood samples from the patient. So even though blood samples can cannot provide I can provide rather insights into disease in the gut, though they don't offer a complete picture of the gut environment. However, certain hormones and metabolites in the blood uh, can show how or what is the condition of the gut, like insulin, glucose, which can act as biomarker. So after getting the blood sample, uh, the doctor and then there are processes involved in the hospital na ibigay yung sample sa biotech sa biotech, and then the I medtech rather, the medtech, and then uh the blood sample will be cultured, and then after having a pure culture, it will be subjected to further analysis like PCR, etc. And then uh, this will be ano to? Dyes will be utilized to view these microbial communities present in the blood. And then the doctor uh, will talk to the patient regarding its condition, kung uh, ano yung mga diseases na meron siya, if meron man in its digestive system. And the doctor will offer services to the patient, which could include direct surgery or hospitalization. So you can see the difference between the shift of using biomarkers and cell stains from conventional method, which is the exploratory surgery. Um, Next is, so here are the advantages of using biomarkers and cell stain in modern diagnosis. So first, early detection. So it helps detecting diseases at an early stage, often before clinical symptoms appear. Specificity and sensitivity. So it allows the identification of particularly diseases or microbial communities with greater accuracy without open surgery. Next is non-invasive. So this biomarker test can oftenly less invasive compared to the traditional diagnostic methods like biopsies or surgery. Um, Next is rapid results. So it can provide quick results, which can be crucial in time-sensitive conditions. Next is personalized medicine. So it allows a more personalized approach to treatment, as they can reveal genetic or molecular factors that influence how an individual might respond to certain therapies. And lastly, quantitative data. So these staining methods, accompanied with biomarkers, can provide quantitative data that can help in monitoring disease progression or treatment effectiveness. Oh, na-vlog. And there are few disadvantages in using this biomarkers and cell stain. Of course, the equipment and reagents required for biomarker testing and advanced cell staining methods can be expensive, making them less accessible if hindi advanced yung hospital, or you're just an ordinary person na gustong itry ito, ang cell stains and molecular markers. And then it it is complex. So these methods may require highly trained personnel and specialized equipments. Next is, it may be false positive or negative, since Bioma- even though biomarkers are specific, certain biomarkers might be not present in healthy individuals, leading to false positives. So also, biomarkers might not be detectable in the early stages of disease, which could lead to false negatives. So next is limited biomarkers. So the effectiveness of biomarker-based diagnosis is restricted to biomarkers that have already been identified, potentially missing out on unknown or rare biomarkers. Next is interpretation challenges. So, biomarkers levels can be influenced by various factors such as medication or other underlying conditions. Lastly, there are ethical concerns about this usage, since it uses genetic or molecular biomarkers. Uh, there is a concern of privacy or data security and potential misuse of sensitive genetic information. So let's move further. Um, naglag. Through, okay. Lag. So after the successful breakthrough in the medicine on utilizing this biomarker and cell stains, researchers see it is can be valuable in detecting diseases in plants and even in soil. So here's an example in which healthy tomato, you can see the difference in an infected plant which has Fusarium wilt. So you can see that it is yellowish and the leaves is dry na siya, it's dead, and has stunted growth. So in infected plants with Fusarium, uh, it shows symptoms like wilting, yellowing in leaves, and eventually leads to death of the plant. So uh, behind this Fusarium wilt is the agent named Fusarium oxysporum, which is a soil-borne fungal pathogen. So how is it importance in the agriculture? Let's find out. So Of course, this fusarium wealth is um, indication that the soil and the plant is unhealthy. So researchers don't want the ano ba to? Researchers utilize DNA based biomarkers to detect fusarium wealth in tomatoes. So first, getting an fusarium infected tomato with fusarium wilt, a tish, a leaf, or an roots um, to, will be cultured. I'm kukunin yung sample tissues from the leaf or the roots, and then the DNA will be extracted. Lang nag-load ang picture. So, the DNA will be extracted from this root or leaf, and then subject to PCR for amplification of the target sequence. So the target sequence is the Fusarium oxysporum. And then subject to gel electrophoresis of PCR results to confirm if merong Fusarium wealth in these tomatoes, and this could lead to the researchers having action to this problem. They could use pesticides uh, uh, to inhibit the growth of this Fusarium wealth in the plant. So here are few advantages of using biomarkers and cells in detection of diseases in the plant. So first is early detection. So same as the a while ago discussed, it allows early identification of diseases, but here it is the plant diseases. So before visible symptoms may appear, so it enables prompt action to manage and control the diseases. Next is non-invasive. So many biomarker-based methods are non-destructive to plants. So meaning the plant doesn't have to be harmed or sacrificed to test for diseases which preserve its health or further studies. So when we say non-invasive, it means not harmful and doesn't alter the sample. So next is the specificity. So biomarkers are highly specific to certain pathogens or disease states, making it easier to accurately detect specific diseases or infections without interference from other factors. Next is quantitative. So biomarkers can often provide quantitative data such as level of pathogen presence, which could help in assessing the severity of the disease or monitoring its progression over time. Next is precision. So, biomarker detection can offer high precision in identifying pathogens, allowing for targeted interventions rather than broad spectrum treatments that may harm non target species. And lastly, it is rapid and cost effective. So in many cases, using these biomarkers can lead to faster and more cost effective disease diagnosis compared to traditional methods like culturing or microscopic examination. So in this case, the biomarker is the sequence of the fusarium, which was detected in the plant. Next, there are few disadvantages in using biomarkers. So first is complexity of the detection. So some biomarker detection methods like PCR or LISA might require specialized equipment and trained personnel, which makes them inaccessible to resource limited areas or non-experts. Next is it is false positives or negative sometimes. So biomarker based tests can sometimes produce false positives, which indicates that disease when even though it is not present. So uh, or false negatives, which fails to detect the disease, which leads to incorrect conclusions or unnecessary treatments. So how does this false positive achieve? So uh, maybe there are interactions with related biomolecules na related to the biomarker, and then this could lead to false positive na even though hindi na-hit yung presence ng specific biomarker, and then false negative, maybe uh, low amounts of the biomolecules kaya hindi siya na-detect ng biomarker. So which leads to incorrect conclusions. Next is limited to specific pathogens. So many biomarkers are specific to certain pathogens, which means they may not work for all the diseases. And a plant might need to be tested for multiple biomarkers to rule out various diseases. Lalubat. Wait lang. Ito na to. So, sensitivity, some biomarkers may not detect pathogens in low abundance or early in the infection process, which could also discussed a while ago, can lead to false negative, making it harder to detect disease in the early stages or when they are at low levels in the plant. So, next is cost of equipment and reagents. So, some biomarker assays like PCR, which will be discussed later, can be expensive due to the cost of reagents and equipment required, which may limit their widespread use in some regions. So, lastly, is environmental factors. So, environmental conditions like soil quality, humidity, and temperature can sometimes interfere with the accuracy of these biomarkers, potentially leading to inconsistent results. Okay. So, so here are some biomarkers. As discussed, the gut microbiome. So the gut microbiome possess good and bad bacteria. So this good and bad bacteria must be balanced. So 80% of the good bacteria must be present, such as this lactobacillus, and bifidobacterium, and many more. And then bad bacteria must be in 20%, which includes the Salmonella, E. coli, Staphylococcus, etc. So changes in this microbial communities in the gut could lead to digestive system diseases or complications. And then, so here, the biomarker is the microbial communities. Next is phytochemicals in plants. So here is an example. It's a plant. Ano to na plant? Uh, a plant when some plants when stressed or facing a threat release phytochemicals, which for example is alkaloids, which prevents herbivores from uh, consuming them. So the biomarker here is the uh, phytochemicals, which indicates that the plant might be in stress or facing a threat. Next is staining. In staining, dyes are used to view the cellular components or cellular structures of microbes. So here, the dye is the biomarker. So let's dig deeper in the stains. Hala, oy. So in cell staining, several staining methods are suitable for quantifying and identifying microorganism in natural samples. So cell staining is an important technique in microbiology and cellular biology used to enhance the visibility of cells or specific cellular components under microscope. So some purpose of staining is to differentiate cell shape, size, and structure. Next is identify cell types and even detect the presence of certain biomolecules that act as biomarkers. So this cell staining can help in characterizing a microbial community in samples. So there are different types of stain. So I have three here to show you. First is simple stain, which uses a single dye to color the entire cell. So as you can see, uh, the cell um, is stained with methylene blue. So methylene blue. Since methylene blue is utilized, the cell appears blue in color. So this could be useful in studying the entire cell structure. And next is differential stains. So in differential stains, it utilizes multiple dyes to, to distinguish between cell types or structures. Famous an example of is gram staining, which differentiates gram positive and gram negative. So how is gram staining different? Do this? So through the component of the different cell wall components. So we all know the gram positive have a thick peptidoglycan, right? So it only allows the purple stain from the crystal violet to integrate to its cell wall. And next is in the gram negative bacteria, uh, which have which have a thinner cell wall and have an outer membrane composed of lipopolysaccharide, which retains the red or pink color of safranin. Um, Next type of stain is the special stain, which targets specific cellular features, which highlights the unique cell components. Example of this is uh, in the spore staining, which uses the malachite green stain to detect spores in bacterial cells. So we will know that endosspore indicates that there is the microbial community present is resistant, creating ano to, spore structure. So here, the the green the green indicates the spore in the of the bacterial community. So let's dig deeper in cell staining. So oh, uh, in staining, fluorescent staining is also used as a biomarker. So this fluorescent staining uses specific dyes that glow when exposed to specific wavelengths of light, such as UV light or blue light. So they play a crucial role in characterization of microbial communities by enabling the visualization of specific biomolecules present within microbial cells. So these microbial staining dyes, microbial dyes, fluorescent dyes, uh, were used to stain cell membranes or proteins, which allows researchers to observe cellular structures with high precision under fluorescence microscopy. So we can only see this glow through fluorescence microscopy. Next, these stains facilitate the identification and differentiation of microorganisms with complex microbial communities by highlighting distinct cellular features such as nucleic acid, proteins, or even the metabolic activity of the cell. Fluorescent staining not only aids in identifying individual species, but also allows analysis of their uh, distribution and interactions, which may provide valuable insights into their diversity and important roles in various environments. So how is this fluorescent staining dyes used? So first, we have here a fluorescent dyes. Example here is a DAP eye. So the microbial sample will be stained with fluorescent dye and observed in fluorescence microscope. So you may wonder if this microbial sample can be seen with a fluorescent microscope. Excuse me. No, because only fluorescent microscope allows or acts as the source of the UV light. So even though you have this sample in a slide stained with fluorescent dye, it will not glow. So you need to use this fluorescent microscope to view it structure with a glowing effect. Next, under viewing, sorry, this some microbial sample under fluorescent microscope, you will this allows the visualization of the cell stain. So here, example here is the uh, endothelial cell stains with the DAP eye. So here, the blue dye indicates uh, the DAP eye. because the dap eye stain is blue or fluoresce blue color. Um, so in the next slide, we will explore this dap eye. Excuse me. So samples of fluorescent stains, again, the dap eye, which have cyupac or 6-diamino 2-phenylindole. So it is commonly used for characterizing microbial communities, particularly for DNA staining and identification of cells under fluorescent microscopy. So it only binds to a minor grove of double-stranded DNA, then emits blue fluorescence when exposed to UV light, which the source of the UV light is in the fluorescent microscope. And then the intensity of the fluorescent corresponds to the amount of DNA in the cell. So it makes the DAP eye an effective method for estimating cell numbers. So uh, the more intense the fluorescence of this DAP eye is proportion to the amount of DNA present in the sample. So here is chemical structure of the DAP eye. So here, you can see here the minor groove and the major groove of DNA. So as discussed, the DAP eye binds to the minor groove, so it binds here. So let's dig deeper in the next slide. So here, as I said a while ago, the DAP eye fluoresced blue blue. Here sample is a seawater without marine broth. So maliit lang yung DNA na kuha or na stained. And when subjected to marine broth, uh, which give a source of nutrients to the cells. So more this leads to more DNA. As you can see, it has higher amount of DNA. The beak with seawater plus marine broth have uh, a lot DNA compared to the seawater without the marine broth. So this figure only shows how DNA can be viewed through the utilization of this DAP eye. So again, the DAP eye fluoresces proportional to the amount of DNA present in the sample. Okay. So here are advantages of using this DAP eye. So first, it is broad staining since it only stains cells with DNA. So making it useful for visualizing the total microbial population in a sample regardless of species. So regardless of the species, it will stain cells with DNA. Next is quantification. So uh, DAP I's strong DNA binding allows for the quantification of microbial biomass as fluorescence intensifies, which correlates with DNA content. So I said, the more the fluoresce, the more DNA in the sample. Next is simple and cost effective. So DAP eye is inexpensive if you are a researcher in need of this stain, and it is easy to use. which makes it accessible for routine use in microbial community studies. So, as according to my recent research, kanina lang siya. This DAP-I cost 3,641.36 in Philippine peso. So, if interested kayo bumili, there are sites for this. So, now, lastly, its compatibility with fluorescence microscopy. So, DAP-I works well with fluorescence microscopy, which allows for detailed visualization of microbial cells in environmental samples. Uh, because not all fluorescent stains are compatible with fluorescent microscope. Uh, so here are a few disadvantages of this DAP-I stain. One is non-specific. Since it stains all DNA, you cannot differentiate between live and dead cells. So this limits its ability to provide insights into the microbial activity. And second, limited specificity. So DAP-I does not provide information on specific microbial groups or their functions, which requires additional methods like FISH for more detailed characterization, which will be discussed later on. Uh, Next is potential cytotoxicity. So high concentration of DAP-I can be toxic to cells, so it must be uh, done with precautions. So it can potentially affect the viability of the microbes, especially in live cells studies. So lastly, it's UV light requirement. So since DAP-I requires UV light for excitation, uh, this can cause photo bleaching and cell damage, which limits its use for long-term studies. So next, next dye is the acridine orange or the AO. So this dye is used in studying microbial communities to stain both the RNA and DNA in cells. So AO or the acridine orange binds to the RNA, which gives off an orange fluorescence, while so which this binding and the fluorescent specificity, which is the orange, helps identify cells that are more active and have higher RNA levels. So this means that the cell is actively undergoing transcription, and then when it binds to DNA, this acridine orange shows a green fluorescence, which helps spot DNA-rich cells. So this makes acridine orange useful for detecting active microbes in sample. So since active cells usually have more RNA since they are actively transcribing. So by staining both types of nucleic acids, this acridine orange help researchers understand the activity and number of microorganisms in different environments. So again, in DNA, uh, this stain binds to the double-stranded DNA, and this causes to emit green fluorescence when excited by light. Uh, and while the in RNA, it binds to the single-stranded RNA, which particularly in more than one form. So uh, it tells that the cell is transcribing. Then when it binds to the RNA, it emits orange fluorescence. Mm. So here is an image. So as you can see, there is a uh, orange and green fluoresce. So here as the sample used is an adhesive tape stained with acridine orange. So as I said a while ago, the the orange fluoresce tells you the RNA uh, cells, while the green tells the DNA specific cells. So that's it for the acridine orange. So here are a few advantages of using this AO dye. First is dual nucleic acid staining. So it stains both RNA and DNA, which allows the researchers to distinguish between RNA-rich, which is active cells, and DNA-rich, which concludes an inactive cells in a microbial community, which provides insights into the microbial community if the cell is actively transcribing. So next is detection of metabolically active cell zones. Since acridine orange stains RNA in active cells, it particularly useful in identifying metabolically active uh, microorganisms which have higher RNA content due to ongoing protein synthesis through transcription. So of course, my transcription, my translation. Next is quick and easy to use. So this acridine orange is relatively simple to apply and does not require complex procedure, making it a convenient option for routine microbial community analysis. And next is its fluorescence microscopy compatibility. So it can be visualized under fluorescence microscopes, which allows for detailed examination of microbial cells in the environmental samples. Again, not all dyes, not all fluorescent dyes are compatible in fluorescent microscope. So lastly is it is cost effective. So this acrylic orange is inexpensive if you have scholarship, that is. If you are a researcher, I think this amount 2000 to 38.94 centavo Philippine peso per 50 mg is less expensive for you. So it is widely available, and which makes it a cost-effective tool for microbial community characterization. Uh, Even though there are advantages, there are also disadvantages in using acridine orange. So one is non-specific staining. So acridine orange, since it stains all RNA and DNA, you cannot differentiate uh, between different microbial species or provide specific information about microbial function or identifies with the identify identities within a community. So it needs further analysis. Next is limited information on cell viability. So it only identifies RNA-rich active cells. It does not distinguish between live and dead cells. So additional viability stains are needed to assess cell health or to determine if the activity is linked to living cells. So it can be used together with other fluorescent dyes to assess if it is alive or a dead cell. Next is potential for photobleaching. As the same with the DAP eye, it uh, it can be subjected to photobleaching, meaning that its fluorescence fades over time when exposed to light. So this could reduce the ability to collect long-term data or perform time course experiments. So may time limitation ang acridine orange, just like the DAP eye. Next is overlapping fluorescence zones. Since acridine orange binds to both RNA and DNA, there can be overlapping fluorescence. uh, mm, especially if the RNA or DNA contents is high in some cells. So this can sometimes complicate the interpretation of results kasi nag-overlap yung fluoresce na binibigay nila ng sample specifically. So lastly is the toxicity to cells. So just like DAPI, high concentrations of acridine orange can be toxic to cells, which potentially alters the microbial community being studied, especially in live cell assays. So this should be done with cautious, specific amounts, and ayun. Let's move further to another stain, person stain. Naglag lang. Hala. Okay, pamudara. Nainom sa tubig. Next is cyber green. So I guess some of you are familiar with this fluorescent dye. So it is used for detecting DNA and various microbial and environmental studies. So it binds specifically to double-stranded DNA by inserting between the base pairs, which produce a strong green fluorescence when excited by light such as UV light or blue light. So this property makes cyber green an important tool for highlighting microbial DNA environmental samples such as water and soil, which are sensitive. Uh, it is commonly used to visualize and quantify microbial communities in these samples. So it is highly sensitive, allows researchers to detect even small amounts of DNA, making it effective for analyzing natural habitats where microbial populations are often present in low concentrations, such as in seawater, in which DNA is easily degraded and present in small amount. And because of its strong and specific binding, this dye is particularly useful for assessing microbial presence and biomass in studies focusing on water quality, soil health, and microbial community. So let's dig deeper with this dye. Naglag. So here is an image showing the fluorescent of the cyber green. So this cyber, from its name, it fluoresces green under UV light. So here, bacteria and viruses stained with cyber green. So the sample was taken from the Bay of Villefranche Francis in France. So these large bright dots are bacteria, and smaller dimer dots are viruses. So it is from Bay. So it simply stains the DNA present in sensitive samples such as water. Uh, so here are a few advantages of using cybergreen. First, it is highly sensitive. So as I said a while ago, it can detect even low amounts of DNA, making it ideal for samples with small microbial populations such as in natural water or soil. Next, it is suitable for live and fixed cells. So unlike many other dyes, uh, cyber green can stain both live and dead cells. So just like the AO and mm, DAPI. So it allows researchers to study microbial communities without killing the cells. So there's all real-time quantification. So once the DNA, the cell is stained with cybergrain, it increases with DNA quantity. So it is useful for quantitative PCR to measure microbial DNA levels accurately in real time. So this cyber green inserts in the DNA, which increases this DNA quantity. So unlike the DAP-I, so sa minor groove lang siya gabind. So it doesn't increase the quantity. quantity of the DNA present in it. Unlike the cyber green, when binds to the DNA, uh, it increases the DNA quantity present in the cells and the sample rather. So next is distinct green fluorescence. So the bright green color helps in multi-stain experiments, and it doesn't overlap unlike the AO much with other fluorescent stains. So lastly, wide range of applications. So cybergreen is effective in environmental studies, particularly for assessing microbial DNA in soil, water, and other complex samples, which are sensitive. Hello. So if it has advantages, it also have disadvantages. So few of this is non-specific to microbial types. So it just like the DAPI, it binds to double-stranded DNA. Uh, so it doesn't differentiate between microbial species or types. So additional methods are needed for more specific characterization. So photo bleaching. So just like the AON, the DAPI, it can lose fluorescence over time with light exposure. So it has limitations in using it for observation. Next is DNA only. So it does not bind to RNA unlike the AO. So it misses informations on metabolically active cells that have high RNA content, limiting insight into cell activity. So next is potential toxicity. So just like um, the other dyes, fluorescent dyes, it may be toxic to live cells when uh, higher concentrations are utilized. So this can interfere with studies that require observing live microbial communities over time. So background fluorescence is the last disadvantage. Cyber green may sometimes show background fluorescence, which could complicate accurate interpretation of microbial DNA levels. So parang may background daw siya. Uh, fix. So last fluorescent dye is the backlight bacterial viability stain. So um, unlike the previous dyes, they cannot differentiate the live and dead cells. So this backlight can be incorporated together with those dyes to differentiate the live and dead cells. So it has two components, which is the SYT09 or the C29, which stains specifically the live cells by entering their membranes and binding to DNA, which emits green fluorescence specifically. And the next component is the propedium iodide or the PI, which only penetrates dead cells with compromised membranes and binds to their DNA, which emits red fluorescence. So the differentiation of the dyes or relies behind the integrity of the membrane. Since the membrane in the live cell is still intact, it allows the green fluorescence to enter the membrane. While in the dead cells, in which the membrane is disrupted, it only allows red fluorescence to enter the membrane. So these dual staining methods allow researchers to clearly distinguish between metabolically active and inactive dead cells in a sample. So these provide valuable insights into the health and viability of microbial communities in environmental samples like water and soil. So by assessing the ratio of the live to dead cells, scientists or researchers can better understand the complexities of microbial populations, which includes the responses to environmental stress or changes in habitat conditions. So this backlight bacterial viability stain together with the other stains can be utilized in waste water ito, wastewater tawag dito research, um, for example in uh, researchers studying the wastewater want to know if the wastewater disinfectant they use is effective in killing certain microbial pathogens or cells. So this backlight bacterial viability stain can be used to see this effectiveness and can further conclude uh, how much concentrations is needed for killing these uh, specific cells or pathogens. Uh, that's it for bacterial viability stain. So, ala, lang. Okay, so here's image showing this backlight bacterial viability stain under fluorescent microscope. So as you can see, the live is, is the green green forest, while the dead is the red forest of the stained microbial species. So again, it can be used in assessing uh, wastewater disinfection. So lastly is the green fluorescent protein as a cell tag in a reporter gene. So it isn't a dye but a gene. So uh, GFP or the green fluorescent protein considered both a flag and a reporter because it acts as a marker or signal that provides specific information about the cell it labels. So why is it a tag? So it attaches to the target microorganism excuse me, and easily identifies them under a microscope since it fluoresces uh, particular color or fluorescence. So it doesn't alter the cell's function but simply marks it excuse me, with a green glow, allowing researchers to track and monitor the specific cells in a community. So it is analogous to a name tag. So at first, uh, excuse me, we use name tags to identify ourselves right in specific conference or in a place where we are unknown. So just like a name tag, wearing a name tag, this could allow uh excuse me. Oh, nain ako tubig de. Uh, this could allow identification of a specific microorganism under a microscope. Uh, JFP as a reporter because it provides information of the cell status or environment. So just like a traffic light. So since it fluoresces a color or a pigment, uh, it tells the researchers about what is happening to the microorganism. Uy, tubig. Oh, okay. Uh, this fluoresce, as I said, this fluoresce tells the researcher what is happening to the specific microorganism. So this fluoresce can be mawala siya, which tells that there is something wrong with the microorganism. So this GMP is inserted into the microbial species through a transformation or any technique such as plasmid, uh, vector, and then researchers can make cells fluoresce green when exposed to UV light. So which makes it visible under microscope. So this fluorescence is oxygen dependent. So it is only effective, mostly effective in aerobic environments. So it is not um, advisable or utilized in anaerobic environments such as sulfur reducing soil with sulfur reducing bacteria rather. So this GMP tag cells allows researchers or scientists to observe cell interactions, localization, and the dynamics of microbial communities. Additionally, there are fluorescent protein variants such as red fluorescent protein, which allows molecular labeling for more complex studies of microbial ecosystems. However, as I said, it relies to uh, oxygen, which is not advisable to be utilized in anoxic environments. So there are variants such as FP1 to FP11, which have unique excitation and emission wavelengths, which offer flexibility for various environmental needs. So this GFP uh, protein, this green fluorescent protein uh, can be present in many uh, many variants in a in a specific microorganism to study its interaction. So here is an example scenario. So So this is how GFP works. So researchers are studying how beneficial bacteria like rhizobacteria colonize the roots of plants and promote growth. So to track and visualize this bacteria, uh, the GFP tagged is introduced to the bacteria. So meaning the bacteria will express green fluorescent protein. So the first the gene is inserted in the bacteria through a transformation or plasmid vector, etc. And then uh, it is inoculated in plant roots directly. So this allows the researchers to observe the colonization of the uh, rhizobacteria in the roots of the plants. And next, it tracks interactions of this rhizobacteria with other microorganisms. Also, the researcher can also uh, differentiate the bacteria from other microbes and cells in the root system, giving them clear understanding of the plant microbe interaction. So, other than this, uh, GFP can also be utilized in tracking the metabolism of a certain microorganism in a sample. So here is an image showing the different variants of the GFP protein. So the FP ones you can see emits this a fluoresce this color. So there are 12 variants of the green fluorescent protein. So each fluoresce different colors. So here in the B uh, figure, the FP5 or GFP flourished a green in green, uh, while in the figure C, This shows how the using the GFP, the the researchers were able to track the activity of the certain microorganism in a specific part of the plant, or such as in its roots or its leaves. So that's all for my part. I hope you learned something.
Good morning. Marinig niyo ako? Wait, nag-echo. Okay, marinig niyo ako? Okay lang. Sorry. Dalawa mo, gamit ko na ano. So, marinig ako? Okay lang? Okay, so thank you so much. So, the next topic that we'll be discussing, wait lang guys, okay, saba sila. Excuse, pwede din mo mag-singit yung report ko. Thank you. Okay, sorry sa disturbance. So, ato. Okay, so the next topic that we'll be discussing is about PCR or polymerase chain reaction, which all of us already know from our bioinformatics subject. Thus, I'm expecting guys, na since fresh pa siya, that we can just breeze through this topic. Next slide, please. Next slide. Next slide. Thank you.
Lang galag.
Okay, so PCR is a technique in molecular biology that allows for the amplification or the rapid replication of specific segments of DNA. So, the target DNA is rapidly amplified through a series of temperature changes and enzymatic reactions, which we will discuss later on. So, PCR can generate millions to billions of copies of a particular DNA sequence, making it a fundamental tool in genomics, biotechnology, and various biological research applications. Next slide, please. Next slide, please.
Wait lang galag.
Okay, thank you so much. So before we delve in on what PCR is or how it's done, let us first discuss the basic principles of PCR. So we already know that the DNA molecule is a double helix, which is composed of a phosphate group, deoxyribose sugar, nitrogenous base pairings, and nitrogenous base pairings, which are the adenine and thymine, and the guanine and cytosine pair. Next slide, please. Okay, so there are key several enzymes and reagents that are fundamental to the PCR process. So this includes the DNA polymerase, which is the enzyme that synthesizes new DNA strands by adding nucleotides to the 3' end of primers during the extension phase of PCR. So we'll discuss about that extension phase later. So the most commonly used DNA polymerase in PCR is Taq polymerase, which was already discussed by the previous reporters, which is a bacterium that is thermostable and can withstand the high temperatures used in the denaturation step. Moreover, Taq polymerase allows for continuous cycling of the reaction without the need for replenishment. And next, we have our primers, which are short synthetic DNA oligonucleotides designed to flank the target DNA sequence. So the primers provide a starting point for the DNA polymerase to begin its synthesis, and their specificity ensures that only the desired DNA segment is amplified. So we have forward and reverse primers here, wherein the forward primer